All right, welcome back everyone to the deep dive. We've got a really fascinating topic to unpack today, and I think it's going to be a really interesting one, especially for anyone who's been following the latest in gene editing.
Yeah, definitely some cutting-edge stuff here and some pretty big implications to think about.
So for this deep dive, we're going to be looking at this breakthrough you flagged coming out of Japan, and it's all about genetically modifying monkeys, right?
That's right, yeah. Specifically, cynomolgus monkeys, which are often used in research because they're pretty similar to us in a lot of ways.
And what makes this particular research so groundbreaking, at least from what I've been reading, is that they've managed to do this without using viruses.
Yeah, that's the key. They've moved away from those traditional viral methods, which, as you know, have always had some pretty big drawbacks.
Right, right. Viruses, as effective as they can be at getting into cells, also come with, a whole set of baggage in terms of safety and whatnot.
Exactly. And this new research, it seems like they've sidestepped a lot of those issues, which is a pretty big deal, if you ask me.
Okay, so before we get into the specifics of how they actually did this, maybe we should take a step back and talk about why genetically modifying non-human primates is so important in the first place. I mean, we already use animals in research, so what makes these primates, so special?
Well, it all comes down to how closely their biology resembles our own. We've learned a lot from studying mice and rats, but when you're talking about complex human diseases, especially things like infectious diseases or neuropsychiatric disorders.
Like Alzheimer's, Parkinson's, schizophrenia, that sort of thing.
Yeah, exactly. You really need a model that more accurately reflects the intricacies of the human body. And primates, particularly these cynomolgus monkeys, they're just much closer to us genetically and physiologically, so they offer a level of insight that you just can't get from rodents.
That makes sense. So using primates as models could potentially lead to more effective treatments for these diseases, right?
Yeah.
Because we're essentially testing them in a system that's much closer to the human body.
Precisely. It allows us to study how diseases progress, test the effectiveness of new therapies, and really get a clearer picture of what's happening at a molecular level.
Okay, so I'm starting to understand the importance of having these primate models. But you mentioned that the traditional methods for genetically modifying them, the ones using viruses, have had some limitations. Could you elaborate on those a bit?
Sure. So the biggest issue with viral methods is that they can be, a bit risky. You're dealing with modified viruses, and while they're designed to be safe, there's always a chance of things going wrong, especially when you're working with primates.
So safety is a major concern. What else?
Well, another limitation is that viruses can only carry a limited amount of genetic material. So if you want to introduce complex genetic modifications, it becomes really tricky. And then there's the issue of screening.
Screening. What do you mean by that?
With viral methods, it's difficult to know for sure whether the genetic modification has actually taken hold until much later in the process. You essentially have to wait until the embryo has developed further, and that means a lot of wasted time and resources if it turns out the modification didn't work.
So you can't really check your work early on. Okay, so that's a big drawback.
Yeah.
So how did these Japanese researchers overcome these limitations? You mentioned they weren't using viruses. What did they use instead?
They used a system called the PiggyBac Transposon system, and honestly, it's a pretty ingenious approach.
PiggyBac Transposon system, that's a mouthful. What exactly is a transposon?
It's basically a segment of DNA that can move around within a genome. Think of it like a jumping gene that can insert itself into different locations.
Jumping genes. Okay, that's a helpful visual. So they're using these jumping genes to insert the desired genetic modifications into the monkey's DNA.
Exactly. And what makes the PiggyBac system so special is that it's incredibly efficient and precise.
Okay, how so?
Well, for one, it can carry much larger pieces of DNA than most viruses, so you have much more flexibility in terms of the genetic modifications you can introduce. And secondly, it integrates into the genome in a very predictable way, which means you can screen embryos much earlier in development.
So they could basically confirm that the genetic modification has worked before the embryo even gets implanted.
Yes. And that's huge. It saves a ton of time and resources and makes the whole process much more efficient.
Got it. So they're using these jumping genes to deliver the genetic cargo, and they can check if it's worked really early on. That's pretty amazing. But what exactly did they modify in these monkeys? The article mentions something called a transgene.
Right. A transgene is simply a gene that has been artificially introduced into an organism. So it's not a gene that's naturally found in that species.
So they basically added a new gene to these monkeys.
Exactly. And to confirm that this transgene was actually working, that it was being expressed and producing its protein, they used what are called reporter genes.
Reporter genes.
These are genes that produce easily detectable signals, like fluorescence. So in this case, they used two different reporter genes, one that produced a red fluorescent protein and another that produced a green fluorescent protein.
So they could literally see if the transgene was being expressed by looking for these fluorescent signals.
Yes. And what's really remarkable is that they found these fluorescent proteins in all the tissues they examined, including the germ cells, which are the cells that produce sperm and eggs.
Wow, so this means that the genetic modification could potentially be passed down to future generations.
Exactly. It indicates that the modification is stable and heritable, which is essential for creating consistent and reliable animal models for research.
That's incredible. So they've effectively created genetically modified monkeys that can pass these modifications on to their offspring. What are the implications of this for studying human diseases?
The potential is huge. Having these genetically modified primates that can accurately mimic human diseases opens up so many new avenues for research. We can study diseases like Alzheimer's, Parkinson's, HIV, really any disease where primate models are more relevant than rodent models.
It's like having a much clearer window into how these diseases work in humans, right?
Precisely. And that could lead to more effective treatments, new diagnostic tools, and a much deeper understanding of these complex conditions.
That's really exciting. But the article also mentions that while the transgene was consistently integrated into the genome, the level at which it was expressed varied across different tissues. And they talk about the role of promoters in this. Can you explain what promoters are and why they're so important?
Sure. Promoters are basically regions of DNA that control when and where a gene is turned on or off. Think of them like switches that regulate gene expression.
So even if the transgene is present in a cell, it might not actually be active if the promoter isn't turned on.
Exactly. And the researchers emphasize that for future applications, it'll be crucial to carefully select promoters that are specific to the tissue they want to study. For example, if they want to study a brain disorder, they'll need to use a promoter that's active in brain cells.
So it's not just about getting the transgene in there, it's also about making sure it's active in the right cells and at the right levels.
Precisely. And that level of control is what makes this PiggyBac system so powerful.
Got it. So what's next for these researchers? What are they hoping to achieve in the future with this technology?
They have some really ambitious goals. They're working on ways to introduce multiple genes simultaneously, which would allow them to create even more sophisticated disease models that reflect the complexity of human diseases.
That's amazing. So they could potentially create primate models for diseases that involve multiple genes.
Exactly. And they're also exploring ways to control the timing and level of transgene expression even more precisely. It's all about gaining greater control over how these genes are regulated.
And they're looking into epigenetics as well, right?
Yes, epigenetics is a really exciting area of research. It refers to changes in gene expression that don't involve alterations to the underlying DNA sequence. So it's like adding another layer of control on top of the promoter.
So by understanding epigenetics, they could potentially fine-tune gene expression even further.
Exactly. And that could have huge implications for understanding and treating a wide range of diseases.
Wow. It sounds like they're really pushing the boundaries of what's possible with genetic engineering.
They are. And I think this research has the potential to revolutionize the way we study and treat human diseases.
Absolutely. It's a real game changer. And for anyone who wants to learn more about this groundbreaking research, the paper is titled Nonviral Generation of Transgenic Non-Human Primates via the PiggyBac Transposon System by Masataka Nakaya and colleagues. It was published in Nature Communications on March 24th, 2025. We'll make sure to include the full citation in the show notes.
This research is a fantastic example of how overcoming a technical hurdle can open up entirely new possibilities for scientific discovery. It really highlights the power of innovation and collaboration in advancing our understanding of human health.
So to sum up this deep dive, we've learned that this new method for genetically modifying primates using the PiggyBac Transposon system represents a significant leap forward in disease research. It offers a safer, more efficient, and more versatile approach than traditional viral methods.
And the ability to create these genetically modified primates has the potential to revolutionize the way we study and treat a wide range of human diseases, especially those that have been difficult to model in other animals.
It's an exciting time to be following this field, and I can't wait to see what discoveries emerge from this research in the years to come. What are your thoughts on the ethical implications of all this? Does this level of genetic control in primates raise any concerns for you? And what about the potential societal benefits? Could this technology one day lead to cures for diseases that have plagued humanity for centuries? We'd love to hear your thoughts. Until next time, thanks for joining us on The Deep Dive.
Thank you.
And remember to subscribe to stay up to date on all the latest scientific breakthroughs.
We'll see you next time.